model different scenarios, climate change scenarios especially. So our project team consisted of 6 partners, and we had the pleasure to coordinate this project. And we were mainly ecologists, biologists, hydromorphologists, morphologists, hydrologists, and some hydraulic engineers in the project. So what was the main idea? The main idea was that reach scale restoration and reach scale morphology in biota is influenced and affected by pressures at different spatial scales. And so it does not only depend, biota doesn't only depend on the local hydromorphological conditions, but also, of course, on things like migration barriers, source populations, like catchment scale pressures like diffuse source pollution and point source pollution, and also it will depend on global pressures like climate change. So what we do with the restoration is that we usually apply the measures at a reach scale, and in some projects we consider catchment scale processes or pressures, but only in few projects. So what we know from other studies is that these large-scale pressures like catchment land use have a statistical significant effect on reach-scale biota. So in this figure on the left you can see that the share of urban land use in the upstream catchment is limiting the ecological status of invertebrates. And here on the right you can see that the share of the urban land use in the upstream catchment has a strong effect on especially the sensitive taxa. So we did know this, but of course, because it's a statistical analysis, it's difficult really to deduce any kind of causal relationship. So that's why in the impact project we asked the question, what is the relative importance of these different pressures on biota or for biota, and which is really the bottleneck for biota? And we had a focus on climate and land use change as pressures, but also the focus was on restoration issues, because this project also wanted to ask the question, what can you expect from reach scale restoration? Which kind of biological effect can you expect, given that there are other pressures at larger spatial scales which still remain? So what we did is to couple different models which describe different processes at the catchment river network and reach scale, and we did model then different scenarios, different climate and land use scenarios, and we tested the whole approach in 3 different catchments, and in all of these catchments we had one near natural reach as a kind of analog for a restored reach, and tested our modeling approach there. So these are our 3 model reaches, and in this presentation I would like to focus on the Träne study reach, which you can see on the left side. It's a sandbed river in northern Germany, and it looks quite the same as many restored meandering rivers in Germany and across Central Europe. And the scenarios we did investigate was first of all a climate change scenario, and here we used data from the so-called STAR climate change scenarios. These are regional climate change data which were derived from global climate change models by statistical downscaling. And we had one baseline scenario where we assumed no increase in temperature, and a rather pessimistic climate change scenario where we assumed an increase of temperature up to 3 degrees. So you can see it's a rather pessimistic one. In addition, we had some land use scenarios which were developed by one of the partners, and it's important to note that in this specific catchment of the Träne, we already have a very high share of corn as an energy crop in the baseline scenario. And we had then 3 different land use scenarios. The one we call food, where corn even increases more. Then there's an alternative energy crop scenario where we have alternative energy crops, of course, as the name says. And we have a best management scenarios, which is the best from an ecological point of view, where we also have a higher share of grassland. So this is a kind of shocking slide because it shows the model flow chart as a whole. And as you can see, it looks rather complicated, but actually I think it's quite simple if you understand the different models and how they are linked. And the rest of the presentation will be about explaining this model flow chart and to illustrate it using the Träne as a case study. So I would like to say, don't panic. I think after this presentation you will be familiar, and hopefully I will be able to deliver the message what this flowchart means. So at the core of the modeling approach, there is an eco-hydrological model. And as I think you probably know, is that these eco-hydrological models are widely used in ecology and hydrology. And as an output, they model the discharge, but also water quality aspects like nutrient loads and fine sediment loads. So in our project, we used the SWOT model, but just because it was available to our partner, so no specific reason about this. And the output are these kind of hydrographs, as you can see here in this figure. So daily value for the discharge. Now it's not really useful and not feasible to really model the habitat conditions and biota for each day in this hydrograph. So the first step here, an important step, is to choose morphologically and ecologically meaningful hydrological variables. And the ones we did use is the mean discharge and the extremes, so some value for the low flow and some for the high flow. And we calculated these value not for the whole year, but for as monthly values. So we considered seasonality. But actually, if you've got other research questions, you can also use other hydrological variables you are interested in this modeling framework. So these are the results for the Träne study catchment. You can see here 3 graphs which show the monthly values for the low flow, the median flow, and the high flow for the baseline scenario and the climate change. And if you compare the blue boxes and the red boxes, you can see that especially in the late autumn there is a decrease in the discharge predicted by the SWOT model. So this is the effect of climate change, which is predicted in the Träne catchment by this eco-hydrological model. So the second aspect we were interested in is water quality. And here you can see 2 graphs, one for total phosphorus and one for nitrate, which gives the days above a specific threshold value which is ecologically important. And you can see in the up graph that all bars have the same height, so there's not much difference between the different scenario runs. But in the lower part of the graph you see for nitrate that for the scenarios the nitrate values differs. But still, all the values are positive. So still, also in the best management scenario, which is given here as the green bars, there are some days per month which are above the critical threshold. So this means that in these land use scenarios, there are some differences which are ecologically meaningful and which can be interpreted in the following steps. So after this eco-hydrological model, we have a 1D hydraulic model in our model chain or model flow chart. And I'm sure you are familiar with this 1D hydraulic model, which simply gives a so-called rating curve, so a relationship between the discharge on the x-axis and the water level at the y-axis. And this is the water level at the downstream end of our study reach. So this model links the catchment scale process, our modeling of discharge, to our study reach.
So Joachim, did you see there's a question from Hans there? He's asking if the nitrate is in NO3 or N.
Yes, the nitrate is NO3 in this case. So we also did model ammonium, but it's not total N, it's NO3. So the next step actually would be to have this discharge and water level as an input for a 2D hydrodynamic model. But in impact we had one aspect which we also modeled in detail, and this is the effect of discharge changes on the morphology. So we asked the question, are these discharge changes large enough to change the morphology, which of course in turn would affect the biology. However, there is not really one single model, physical model, which can be used to predict the long-term morphological changes, morphodynamics of a river reach. So we used a stepwise approach in impact, and we started looking at the channel geometry using kind of rather simple but well-established empirical models and regime models. And for the Träne case study, we found that there will be a very small change in the bankfall discharge, which is connected to a very small change in the channel geometry. So second, we looked at the channel plane form using a meander migration model, which is called meanders. And we investigated the influence of these discharge changes on the channel dynamics. And here we also found that the changes, for example, in the migration rates will be very low. So no really significant effect on morphology in respect to these two aspects. But we found some significant differences in the channel bathymetry, where we used depth 3D as a model, and the equilibrium bathymetry, which results from the modeling, really differs from the map bathymetry. But after some discussion, we are quite sure that these differences are probably due to the aquatic vegetation which we have in the Träne reach. So there are macrophytes in the reach, and they probably stabilize the channel bed. And because these macrophytes and their effect on morphology is not included yet in these kind of morphodynamic models, we do think that this is just a kind of modeling artifact and not really a difference which is due to the differences in discharge. So that's why for the following steps we simply use the channel bathymetry that we have mapped in the field, and this together with the discharge and the water level were the input variables for the 2D hydrodynamic model. I think you are probably also familiar with these kind of models. So the input here were the discharge value, the monthly values for the three different hydrological variables. And the output, of course, are hydraulic habitat conditions, so depth, averaged flow velocity, depth, and also things like shear stress. And this is a kind of summary for the three different hydrological variables and a comparison between the baseline and the climate change conditions. And I added this dashed black line, which gives the critical shear of the bed material. And you can see that most of the channel bed area is above this critical threshold, also during low flow conditions. So this is a typical example of a meandering channel which has shifting sand most of the time for most of the flow conditions. And climate change will decrease discharge and therefore also decrease the area which has shifting sand. So this is more or less probably a positive effect for biota, because stable sand are better habitats for invertebrates. This is another way of looking at the discharge changes and compare it to the changes in the hydraulics, because this graph or figure shows you the difference between the climate change in the baseline scenario. So all values are negative because discharge is decreasing. Of course, also all the hydraulic variables are decreasing. But it nicely shows that for all the three hydrological variables, we have the same pattern. So same degree of decrease in the summer and autumn month. But it also shows that discharge changes and hydraulic changes do not go fully in parallel. So you've got some months which are indicated by this red box where discharge changes are largest, but the hydraulic changes are less. And this is because the form of the channel, so the channel cross-section, is asymmetric. So you have not a linear relationship between these discharge changes and the hydraulic changes. So after modeling these hydraulic habitat conditions, we now do come to all the kind of biological modeling, which I think is perhaps more interesting because it's less well established in modeling. So first of all, we used habitat suitability models to assess the suitability of these hydraulic and substrate conditions for fish and invertebrates. For the fish, we used a fuzzy logic approach, which, for example, is also implemented in Casimir. You might know this modeling software. And the input here is threefold. First of all, you have to have information about the habitat conditions. You can see here this figure on the left. So on the hydraulic habitat conditions usually. Then you've got in the middle of this figure the so-called membership functions. So all the hydraulic variables are classified with some boundaries, and these boundaries overlap. So it might be that a specific velocity or depth belongs to two classes. And third, you've got some habitat suitability rules, which are usually based on expert judgment. And here you've got to assess each of the specific combinations between the depth and velocity classes and assess the suitability for specific species. So the output of these habitat suitability models are suitability values for each model grid cell. So you've got a computational grid which covers the whole study reach, and you get a value for all the grid cells. And then you can calculate overall value for the whole study reach, which is called the weighted usable area, which is simply the sum of all values weighted by the area of the grid cells. These are the results for the weighted usable area for different groups of fish. You can see here the month on the x-axis and the weighted usable area on the y-axis. And you can see for the large-bodied fish there is a decrease due to the seasonal pattern of discharge in the weighted usable area, simply because the depth decreases. And this decrease is amplified due to the decrease in discharge caused by climate change. A rather different pattern occurs for the small-bodied fish, and this is because for the small-bodied fish they use shallow areas, and therefore with the decrease in discharge and an increase in the shallow areas, the weighted usable area increases. However, obviously in the climate change scenario there is a certain threshold which is passed, and therefore it gets too shallow for these small-bodied fish, and there's a kind of depression during the summer month. So for the invertebrates, our colleagues developed a new model, and they did it for the following reason. In these habitat suitability models, the output is the weighted usable area. And it's difficult to assess which weighted usable area is necessary for a species to really establish in a reach. So you cannot really predict the presence or abundance of the species. And with the habitat evaluation tool, this is possible. So you can predict the presence and abundance of different species because this model is based on empirical data, habitat-specific sampling data. I will not explain the model in detail. This would take another 20 minutes. But the input here are the habitat conditions. So, for example, the share of different substrates. And based on these empirical relationships, the model can predict the habitat presence and abundance. So it's more a statistical model than a really kind of physical-based models. And this is also a main difference between the hydraulic and hydrological model and the biological model. The hydraulic model, so the abiotics, there we really have physical-based models, but for biota we usually have more empirical or statistical models. So now we have assessed the habitat suitability for fish and invertebrates, so we know which habitats are available. But in the impact project we were already also interested in the question, are there species in the catchment which really can colonize these habitats? So we also included dispersal modeling, which is really, I think, the most novel aspect of impact. For fish, there was actually really no dispersal model for fish. And therefore, one colleague, Johannes Radigon, developed the FIDIMO, which is also available as an R package for free. And modeling dispersal consists of 3 steps. First of all, you've got to locate the source populations. You can use SDMs, so these are, again, empirical statistical models, or you simply can use the sampling points. Then you've got to quantify the dispersal abilities, which Johannes did using meta-analysis. And these data are also available in an R package called FishMove. And the third step is that you have the coordinates of the source populations and you have knowledge about dispersal abilities, and you can then implement this in a GIS to really model it for a specific river network. And I would like to mention that we also applied a complementary genetic approach to quantify these dispersal abilities. So the output of FIDIMO are these kind of maps. On the left you can see the source population as points, and because there are many source populations, this looks like little worms. And on the right you can see, after some modeling steps, the probability of occurrence of fish in the model grid cell along the river network. So what the model does is that it uses probability function, which is parameterized using this information from literature, and applies this to each source population, and then calculates the probability of occurrence of the fish for the different grid cells along the river network. So one of the main outputs and results for the sandbed river Treene for our study reach was that the dispersal is mainly limited in our catchment by the number of the source populations and not by the barriers. This is illustrated in this figure because we used 2 different methods to quantify the source population. So we used the SDMs, so this statistical approach, and we simply used the samples, the biological samples that we have. And you can see that these differences are quite large compared to the difference between the blue and the red bars, which are the modeling results with and without barriers. The reason for these small differences is that the source populations are more or less evenly distributed. So if you remove one barrier, there's a low probability that you really have a higher probability for the fish to move somewhere where no source population was before. Sorry. So these results are similar, strikingly similar to results recently reported from an empirical study for invertebrates. So it might be that in the past we looked at focused too much on the barriers, and that it's more appropriate to focus more on source populations. But of course there are a large difference between the species, because some species are migratory species and some are not. And also I have to point out that these results of course are strongly catchment specific. So, but it's interesting to see that there are these similar results in Tonking et al. So finally, after having modeled the habitat suitability, the recolonization potential, and the nutrient loads, we try to find a way to combine these information to do a kind of assessment which of these factors are more important. So for the invertebrates, we found a quite straightforward approach to combine the water quality aspects and the habitat suitability. We simply used so-called dose-response relationships. As you can see in this figure, you've got the dose as the nutrient load on the x-axis and the abundance on the y-axis. And you see how the nutrient loads reduces the abundance, and we use this as a kind of correction factor. So the output of the habitat suitability model, which is the abundance of a species based on the substrates which are present, it is corrected and is lowered by a factor which incorporates the effect of the nutrients. Okay, this was quite straightforward and kind of quantitative approach. For combining the habitat suitability and the recolonization potential, we didn't find any kind of quantitative approach because these two variables have different units or describe different things. So, but we found a way to compare it in a qualitative way. So we distinguished between species which have a high habitat suitability and therefore a higher probability of occurring in our restored reach, and the ones for which habitat suitability is low and which have a low probability. In a similar way, we distinguished between species which will occur in the restored reach over the short term because they have a high recolonization potential, and the ones which will not show up in the first years because there are few source populations or too many barriers which are impassable for these species. So for the fish, I would like to show our results. And again, here in these graphs, we've got the habitat suitability on the x-axis and the recolonization potential on the y-axis for the 0 case, so for the baseline climate change scenario, and for the future climate change scenario. So for the baseline, this figure tells us that there is a small difference in the habitat suitability. So these arrow bars on the x-axis overlap, and these arrow bars give you the monthly values. So there's a minimum habitat suitability, so this is usually the summer month, where we have a low discharge and low habitat suitability. You've got a mean value and a maximum value, and these overlap. And on the y-axis, you've got a recolonization potential, so the share of the river network which can be reached, and there are a larger difference in respect to this. So we conclude from this that in a restored reach, so if you restore a reach in this catchment, the habitat suitability is probably high for many species, but some of the fish will come late because the recolonization potential is limiting the restoration effect. So what about the influence of climate change? So we mainly looked at the minimum values, so the month which has had the lowest habitat suitability. And these red bars here indicate that for many fish species, this minimum value, this bottleneck, decreases. So obviously climate change will have an effect on the habitat suitability. It's a little bit tricky to assess if this really affects the fish, but based on the expert judgment we had in the group, we concluded that these changes will have a significant effect on fish, but that it does not really limit the fish population, so that it does not really question restoration in our catchment. So as a summary, I would like to say that we have developed two new models, one for habitat suitability for invertebrates and one for fish dispersal. And we coupled these models in our study catchments, and we found that it really was a challenge to couple the physical and biological models, because they have these different level of details and are based on physical processes on the one hand side and empirical data on the other hand side. We also found that there are some models missing, especially models which predict organic substrates, which are important for biota, and also important in respect to feedback of these organic substrates like macrophytes on morphodynamics. And we found that it is indeed possible to identify the main bottlenecks for biota using this modeling approach. And for fish, we found that it's in our study catchment, it's mainly the decrease of weighted usable area in autumn, which is important for fish, and also the source populations, which are limited and which limit the recolonization potential. And in contrast, for the invertebrates, it's more the land use change and the present high level of nutrients which are critical, and that the other pressures have comparable minor effect. So this brings me to the end of the presentation, and I would like to thank you for attending this webinar. I'm looking forward for interesting questions, and I also thank the funders and also SCP for organizing this webinar.
Thank you very much, Joachim. We can see that Hans has already got the idea. If you go down to the person with the raised hand, round of applause, show your appreciation for the presentation. Very interesting. And now it's over to you with the questions. And whilst you're working out your questions, typing, getting them in place, your thoughts in place, I have a few questions. One, you talked about deriving the empirical relationships in the reach to put in place the models. And I was wondering how long it took you to do that empirical work. I mean, is that something that can be done quickly for then to move, you'd be able to move on to do the modeling? Is it better to have empirical data already available, or is it something that is, do you imagine, going to have to be obligatory, that if somebody wants to use this system and put this modeling in place, they have to spend a few months or weeks developing those empirical relations?
Okay, so I think you referred to the biological modeling. Am I right?
Slide 30. Yeah.
Pardon? Yeah. Okay. So
slide 30.
Okay. So yeah. Okay. This yes. This is it's based on larger data sets. So at least in Germany, and I know that we have the same situation in other countries, we now have an increasing number of monitoring sites where we have monitoring data and also information on, for example, substrates. And we use these data to establish these empirical relationships. So what the partners did is to based on this large number of samples to calculate mean values for different species on different substrates. So, for example, Gammarus pulex abundance on sand was like 1.2 individuals, and so on. And based on this, it was possible to do this kind of modeling. And of course, you need something similar. And of course, the larger the data set is, and the smaller the scatter, the smaller are the confidence limits, and therefore the better are the modeling results. So I think it's something which is applicable in the next years, because we will have these large monitoring data sets.
Yeah, there's a few questions come through there. Look on the chat.
Okay, I will start with starting with the question of Hans. Yes, I think these empirical relationships, of course, are valid under the conditions that we have now. I think we have to distinguish between different influences. The influence which comes from temperature is probably larger than the influence which comes from discharge changes. As far as I am aware of, at least in Central Europe, there are some predictions, especially for fish species, which show that we will have a shift of the species going upstream because they are sensitive to changes in temperature, but we will have less changes in the invertebrates. So I think for fish it's more a problem than for invertebrates. But I think the range that we have presently in our empirical data, it's large enough to cover the changes that we can expect from climate change. So we already have some sites in very warm areas which are similar to the conditions which can be expected from climate change. There's another one from Jean-Louis Janin. Yes, what parameters should be measured on site? Yes, of course, for invertebrates, most important, of course, are the substrates. And we faced a little bit of problem to model substrate conditions because all the modeling of the sorting of the substrate, especially in mixed gravel sand systems, is not that well established in morphodynamic modeling. So this is kind of problem. So, and this is a field of research where we think also the physical based models should more focus on, so on really the substrate conditions. So I think it's less important to have information, for example, on turbulence, and there is much research on eddy simulation and turbulence, and which I think personally I think is less important for the biota than major aspects like substrate. There's a question, applicability to intermittent rivers. Yes, actually we had an intermittent river, the third one, which is the Quatera in Portugal. We had some funding problems with the Portuguese partner because of the financial crisis. That's why we didn't finish the work there. But I think it would be a challenge to apply to intermittent river, because there you have these kind of pools which remain in the river and which have very different, which have a large influence on the recolonization of the dried channel after the re-wetting. So we already thought about some approaches to apply this modeling framework, but I think it will be a challenge. So it's one has to consider the specific things like recolonization from these pools then. Hans asked about mountain rivers. Yes, I think the conclusions would be very different in mountain rivers. We had one gravel bed river which we investigated, the Selé, which is not really a mountain river, but it's a gravel system. Actually, it's really it was even easier to apply the approach there because we have better morphodynamic models for these gravel bed rivers and a better understanding of sediment transport and less influence of biological aspects like macrophytes there. So I think it should be even easier or is easier to apply there.
That also answered George Meyer's question there as well, but he also adds, are there limits on the model's performance regarding the river size?
I didn't think about this yet. Of course, these are kind of mid-sized rivers because they are still available and you can take samples there. I don't think that in principle it can. I think in principle it can also be applied to large rivers. I can't think of a major problem there. Yes, actually, the question of Antonio Loporto would be fine to test this approach in one of your rivers. This would be great. We still have some investigations running in the Quatera, which is this intermittent river in Portugal, and hopefully we will have some results in about end of this year. But it's difficult to do this without the funding, which has ended yet. Yes, good comment on catastrophic events. No, it's not possible to really include this. It's I think it's a general problem with ecology that we always stick to this kind of mean values, and that we have problems with considering the effect of kind of thresholds which are passed. So yeah, just a response to Hans, because he's mentioning now again the climate extremes and catastrophic events. We were very surprised to see that in this northern German catchment we don't really have an increase predicted in the high flows, in the morphodynamic relevant high flows. This might be a specific case there because it's a catchment which is located between the North Sea and the Baltic Sea. But besides really catastrophic events like 100 years floods, which really dramatically changed the morphology, I wonder if it might be that we overpredict the role of climate change and the discharge changes on morphology, but I'm not sure about this. But it's just a personal feeling that we have not enough information, and I don't know many papers which investigated the effect of climate change, discharge changes on morphology. So we see more catastrophic events like high flows, but will this really change morphodynamics in general? I'm not sure.
I had
okay.
Sorry, I had one other question as well. That you talked about dispersal being limited by the source populations and not barriers, and that was very catchment specific. Did you have similar conclusions from the other case studies? I know you might have it difficult to have the results from the Portuguese case study, but
yes, we have similar results for the Sele catchment, but actually there also the number of barriers is much lower compared to this lowland system. I think if you really have a very heavily degraded system with very few source populations, the barriers are more limiting. But actually, I do think that we have focused too much on the barriers and not so much on the source populations. So, but I must mention that, of course, this refers to dispersal, which is due to kind of, so this is not the dispersal of migratory fish to the sea or back. This refers to the migration that fish do over some few kilometers. So it might be different if you include and are more interested in this migration of the fish to the sea and back. What do I mean by barriers? So dams, weirs, everything which hinders the up or downstream migration of fish and invertebrates.
Okay.
Yeah.
Do we have any more questions? I know Hans is typing there. Is it a question? Okay.
Yeah, so we were lucky in Germany to have, especially for the trainer, that we have a rather good inventory of the barriers, including information on passability, height, fish ladders, and things like this. So that's
okay. It seems that there are no more questions.
A paper on that. Very good question. No, there are 2 papers on this dispersal things and fish, which are out. So this whole dispersal modeling of fish, there's 2 papers. We have not published the modeling framework yet, but we have nearly finished the paper, and the results of the trainer are also nearly finished. So I think in about half a year, I'm quite sure that we will have some results published.
Okay, and this also seems like a question for Hans as well, if he's got any papers on the porosity of barriers from the work done in the UK. We shall ask people send emails via me. I can get you all in touch with each other to pass on information. It's certainly not a problem. If you don't have each other's emails, then certainly ask your requests to myself, s.midgley@oyo.fr, and I can certainly get you in touch with you. There we go. Yeah. Using the system well.
Yes.
But if there are no further questions, we're coming up to the finish of our hour session. Well, I would like to say thank you very much to Joachim for a very interesting presentation. Thank you to the participants for your questions and your interest in the subject. And I wish you all a very good afternoon. And hopefully we will have webinars from the Office of Water in the future. For me, it's a very good system to use, a way of exchanging easily between people across Europe. So good to have your support on these events. And I'll say goodbye to you all.
Yeah, also from my side, many thanks to Stephen for organizing this.